Electrotransfer in microbiology is the technique in which an electric field is applied across a distance of less than 3 millimeters inside of a suspension of cells in a electroporation cuvette test tube. It's a square-shaped one, but it's still a test tube. And it's on the order of several thousand volts. In 3 millimeters. Several thousand volts is enough to create an arc of about an inch. "5G doesn't have the ability to do this because it literally states, when you look it up or test it, how many microvolts per centimeter it is. So, again, homeopathy. What does this do? It increases the permeability of the cell membrane by causing the cell to react to the electric current. Again, cells are in a wet medium and are a wet medium. So in order to get a reaction at them that normally requires an individual atom group called a molecule to trigger something on the cell membrane surface or to initiate active transport, which requires a very tiny chemical reaction, this is like cooking a hamburger using a nuclear warhead. But no 5G, and no, this isn't going to be used on some nanotechnological level when it requires not nanometers but millimeters. That's a bigger size. And microvolts, no, thousands of volts in a 3 millimeter distance. Gene electrotransfer is what you call it when you have the molecule to be transported into DNA for a lab experiment in a test tube to try to get a cell to do something against its will, and is involved in doing things like getting cells to copy things that they wouldn't normally by defeating the outside of a cell membrane. You can sneak it in under Trojan horse method, which is exactly how cells have been tricked into doing things for a long time by viruses or by damage from chemicals, but okay. And electrochemotherapy is when the molecule to be transported are chemotherapeutic agents in a petri dish test to see what it'll do. Again, this isn't a practical application, and no, it's not caused by cell phone antennas or the influence machine or anything like that. There are lots of websites out there that look up a fact. You can't deny this fact because there's a fact that exists. It's reality. There is something called this. For instance, there are RFID tags that are flat or tube-shaped that are used for making cats or dogs or other pets trackable, making your shoes trackable. You may have one in your shoe right now, but people somehow believe that the main drive that they need to worry about is someone attempting to inject them with them, because technically you can get ones that are as size of a grain of salt. And they talk about how they're defiantly not going to take the mark or allow the injection. But anyway, this has nothing to do with the rest of the subject matter, but I thought I'd just get that out of the way. Vaccines traditionally and also recently are made primarily by the following methods. They're called protein vaccines, but that's not the only thing they can do, but primarily proteins, including spike protein. They require the production of a pathogen or a modified version of it, or a reasonable facsimile of it, or part of it. The easiest way to do that is to get a pathogen, a disease, a virus, whatever, it could be other things, and cause them to be reproduced in a container, petri dish, and then from them harvesting protein antigens from the pathogen. This could be pieces of proteins. This could also be other components, but it's almost exclusively proteins. Some foreign component has to be taken from the virus. It could be part of the DNA chain from the virus, although that doesn't seem to be used much or be very useful. Although that can be used in the process of making something else that would be useful. Normally it's based on grabbing whatever it is the virus puts itself into an encapsulation. If you've never seen the cartoonish diagram of a virus, it looks like a little Chinese pagoda tower kind of thing with a couple of legs on the bottom that grip the cell and inject the DNA or whatever it is the virus really is. It is a Trojan horse method. And the primary method we adapt to viruses until they change is that our immune systems react to the outside of these little casings because they're made out of proteins and they're not normal to a human being. But once they get inside of a cell, some of them create a Trojan horse out of pieces of the outside of the cell they've just hijacked and wrapping themselves up in the dead flesh of our cells, so it's harder to kill them. But they almost always modify them, and transferring between humans, especially, they can't do that, because then they would be recognized as not me, but your cells, and my immune system would immediately attack them. We're pseudo, micro, nano, xenophobic stuff going on. So anyway, these antigens, these pieces, can be produced in mass, and the usual method is to generate a bunch of the virus and then go through and kill it and just have the outside casings or something else. It could be, like in the case of COVID-19, it's a spike protein specifically that we could code for, or some other part. It could also be just the capsule itself, or a rough facsimile. It could be also another unrelated virus that we can alter to where it looks like it. It doesn't survive it, but we can alter it. One of the weird cases is doing one of these capsids or capsules or whatever, and altering it by shearing off part of it chemically, breaking it up to where it looks a little like it, and it made a treatment that would cause you to become immune to lots of different things. They also do that for parasites and that sort of thing. It just has to be a facsimile of it. Then we allow an immune system to use it as a practice target to get good at killing it and gain immunity. But these require some mechanism to mass produce this. Usually it's a cell culture that's tricked into copying the antigen by either infecting it or tricking the cell into just copying that piece that'll cause the reaction, the antigen. So the test tube cells can, at that point, directly produce just the antigen. It could be something safe with no virus in it, just the casing of it, or even just one part, like a spike protein, for instance. Then we inject whatever that is produced and cause a protective immunological response. In order to make this 100% pure and completely filtered of any virus component at all, very careful, slow, meticulous work must be done. And that can be nearly impossible. It also makes it prohibitively expensive. But what if you could do that at one stage and then use that being copied by cells? You have cells as well. Why not make use of our own cells? Once inside a human being, a single strand RNA sequence, a messenger RNA almost exclusively, vaccine, in quotes, is a set of instructions that are ignored because they're wrapped in a lipid coating, which can cause a reaction in a person. You try to avoid that, obviously, because you want the immune system and the body to ignore it. Once this lipid coating is ripped off and this messenger RNA is absorbed by the cell, it doesn't need to enter the cell. In fact, it's expedited to going into another component of the cell. Therefore, it can't modify your DNA at all. And functions as normal mRNA. It's usually a copy of normal mRNA that's instructed to do something specific. It instructs the cells to make the vaccine in the first fucking place, since there's no virus involved, and the mRNA is scanned to make sure it's not a component that can be integrated into the DNA, and is a component that has a valid set of codes and passwords and logins and tongue recognition. This ends up being an extremely expensive and then suddenly ridiculously cheap process because you can inject a person with a smaller amount. Or go ahead and inject them with the same amount or even larger in some cases. It requires cold chain distribution and storage to still allow for the cells to be directly produce the antigen or vaccine in your own cells and cause a protective immunization response that is tailor-made for a specific protein. This is limited. If there's a mutation in the disease you're treating, it won't be as effective, but it can be ridiculously safe because it can't physically contain a virus, because if it did, it would disrupt its ability to be copied in the first place. It has the effect of canceling out the process of having a contamination. It also does not require preservation using anything that anti-vaxxers complain about. All of their arguments die because it can't contain a virus component, or it would self-annihilate and not work. Next, it talks your body into doing the production of a vaccine effectively. Next, it uses exclusively cold storage preservation, which means it does not contain antibiotic or antiviral or anti-chemical reactions of any kind. It doesn't contain mercury, thimerosal, or anything else, because that would be useless. Also, if it goes bad, you want it to be detectable, and those would hamper that process. Even hypothetically, they can't be infectious because they don't construct themselves from any active pathogen or an inactive pathogen or an antigen, because they don't contain any of the components. They tell your body to make them instead of making an egg, a chicken egg do it or something, or a petri dish experiment with bacteria. None of those processes are involved, and if you can keep it cold storage, it would have almost no ability to alter itself in any way. But it does require a coating, but not always. You can also have it just injected naked. You just have to increase the dosage, and that decreases any side effects. The mRNA types have only been considered experimental or theoretical, or only for experiments on animals, until COVID-19 happened. This was chosen specifically because, even hypothetically, they're almost incapable of hurting you unless certain responses happen. You must take precautions and exclusions. If an individual has an increased risk of autoimmune reactions, if anybody in your family has an autoimmune disease, don't take this. Just don't. You don't have to. That's a good out. And again, it is voluntary. If you have lupus, for instance, other diseases that can cause an immune system response, or if you have an immune system that is hyperactive only. If it's suppressed, no, that won't be a disqualifying factor. You can ask for a screening to test for strong effects and reactions caused by the protective lipids, the reactionogenic lipids. Make sure you're not allergic to the casing material that they use to encapsulate it. This only happens in higher doses, but you can do a test safely to see if there's a reaction at all. You want no reaction to it. Unlike normal vaccines, by the way, that also include something to antagonize your immune system to get them started. This one is supposed to avoid that, obviously. You could also look to see if you have a tendency towards blood coagulation effects from extracellular RNA testing. You can do this test if you need to. Virtually nobody has these responses, but if they do have them, they will be evident. You can request a short dose or small dosage first, which is what the first dose is supposed to be. It will show a reaction, but it's within safe limits to where you will not overreact. Again, unless you have an immune system problem, or lupus, or some other disqualifying characteristic, or someone in your family does, you probably have nothing to worry about with this, but it is not risk-free. Next, there is something called a DNA vaccine. Double-stranded DNA is different than single-stranded RNA. They've been tested for veterinary use, and when they work by injecting genetically engineered plasmids, not a lipid, containing the DNA sequence, encoding the antigen, as a vaccine that would be produced by the DNA chains. The cells directly produce the antigen, causing protective immunological response, but no DNA vaccines have been approved for human usage at all since, number one, they can also be used for other things like cancers, parasites, and bacteria, and they're being explored as a broad range of other treatments, specifically cancer, because that would affect cancers, specifically. But the other method's cheaper, and they're not going to bother with DNA transfer techniques because it doesn't work yet. Transfer RNA is not messenger RNA, but both of them use uracil instead of thiamine, unlike DNA, which makes them also extra differentiated and prevent interference, damage, or DNA damage. Thank you for watching. Have a good day. Good luck with that.